All right, thanks everyone for coming. My name is Miranda, and if we haven't gotten a chance to chat yet this week, I'm a master's student starting my second year at Utah State University, advised by Dr. Kelsey Graham at that Logan Bee Lab you've been hearing all about. Today I want to talk to you about a third of my project, which is identifying berry crop fidelity in 4 mason bee species using pollen ID, or as I like to say it a bit more succinctly, the proof is in the pollen. Okay, so as a brief background, I know we're all pretty familiar with the bees we have available to us in management. Apis mellifera, or the honeybee, is the one we see the most often. Most of our crops need pollination, and most of our crops get it from this bee. But I think we also know in the room that there are other options. Managed bumblebees, for instance, in the east, and then we have the blue orchard bee here in the west. I posit that there are a lot of other bees we could be using for crop pollination, especially region-specific native bees to the US that could be less impactful than the honeybee. And I think we should be looking at other osmia species because we already know osmia are pretty manageable in a variety of ways. Additionally, orchard crops aren't the only crops out there. I think there's a lot of potential for other species to have good predilections, good phenological matches for other types of crops. In this case, I have just a couple of examples. Osmia bruneri chewing on a strawberry leaf to form its nest, and then Osmia ribiflorus, just its butt, but you can see it digging its head and abdomen deep into a blueberry flower to pollinate and grab some nectar. So managing bees can be quite difficult, managing osmia possibly more so, and managing a brand new osmia is pretty difficult. So there are a couple of steps that the literature has outlined throughout the years to get this done. First, you need to identify a crop that needs better pollination. So if a honeybee is already doing a great job, it's pretty hard to sell research on a new bee. So you need a crop that needs something better. Then you need to identify a bee that pollinates that crop. This is done in a variety of ways, and usually over a long period of time with a lot of studies and a lot of tests. And then 3, you need to assess the benefits of getting that better pollination. So is our new bee doing better than a honeybee would do? So this first step of identifying a crop that needs better pollination, I'm just going to zoom in on a couple of berries. First, the blueberry. Blueberries are super dependent on cross-pollination by bees for good fruit set and good yield. And a lot of blueberry varieties have super strangely shaped flowers. So this graphic just shows that different floral traits make it very hard for certain varieties of blueberries to get good pollination. So depending on how long this bell-shaped flower is, bees can have a hard time getting in and doing what they need to do. Another example is strawberries. Strawberries also need cross-pollination for good yield and quality, not as much as blueberries, but the big difference here is that we can see better pollination when we use mason bees and honeybees together. So this was a study where they had a field with honeybees stocked, but with no mason bees, and then they compared it to a mason bee plus field, and they saw better fruit weight, fruit volume. I am primarily focused on this second step, identifying a bee that pollinates the crop better. Primarily because we've done a few trials in berries with osmia, but very few. And a lot of these were cage trials at the Logan Bee Lab. So for Osmia bruneri, Aglaya, and Lignaria, we know that they like raspberries, but we only know that they like them when they're literally trapped with raspberries as their only pollen source. So we need to put them out in the field and see what they do in a realistic environment like that. Ribiflorus and Lignaria, similar story. They have had field tests, but very limited field tests. And it seems that there's been some trouble getting these bees to be successfully managed out in the field. So we do have people in this room who have tested these things again, but they just haven't published because they haven't been successful. So trying to test these again and create replication is really important. So my objective for this part of my project is to determine if 4 osmia species pollinate berry crops. My basic overall method for doing this is to ID nest pollen provisions. And based on the research I showed you on the last slide, I expect Osmia ribiflorus to collect blueberry pollen, Lignaria to collect strawberry, and Bruneri and Aglaya to collect raspberry pollen. So my study design is pretty simple, and I'm glad Kimball was able to get into the room for this because I give him credit for this awesome nest design. Put these nests out on berry farms and see what the bees do. Pretty simple. Really important aspect of these nests is that we put straws and reeds inside of them. These are nests that I can remove and then sample pollen from. And that's exactly what I do. I let the bees do their thing for a couple weeks. I check them on a weekly basis. And when they've capped their nest, that means they're all done pollinating for that nest. And I take the nest out of the shelter. We have a really cool veterinary x-ray at the bee lab, and I use that to identify exactly where cells are placed within the nest and where those pollen samples are kept. And I can use that to safely extract a random pollen sample from the nest without harming the bee larva. That's the goal anyway. Sometimes I do harm the bee larva. Then I create some pretty simple pollen slides, and I compare them with crop references. A lot of us in the room are using molecular methods for pollen ID, which is great, really useful. This is kind of a faster, cheaper way, and specifically when you know what crop you're looking for, this can be a really simple and easy method to do this. So I have some results to share with you. Just briefly, an overview. I tested currants, blueberry, raspberry, and strawberry in Utah and some of these crops in Oregon as well. And I found that Osmia bruneri was quite attracted to the currant plant. I did find an association for blueberry and Osmia ribiflorus, big question mark on raspberry, which I will explain, and then strawberry and Osmia lignaria. So first for currants, these are some reference images for your reference. I had some of my own as well. This is an interesting one from like an electron microscope, I believe, and I feel that both are important. Because when you look at my pollen, you can kind of see traits of both this just simple fuchsian dye slide and then some of these more interesting traits that you see in the electron microscope. We only had 2 nests for bruneri. This is because currants are traditionally grown in England. We had one Utah site that we wanted to check out, but it's just a very cold weather hardy plant that blooms very early. And Osmia bruneri, in contrast, is like a midsummer flying bee. So they had a hard time coming out for a crop this early. But it was cool that we saw a strong association. So right now I'm working on developing some overwintering methods to try and bring Osmia bruneri out early in the season to get them flying and more hardy to that winter cold. Next up, blueberry and Osmia ribiflorus. So these guys are a story that has been going on for a long time. We've been trying for a while to get this bee to stick. In 2023 in Utah, we had a pretty weak blueberry field that our grower was trying to recover, and it did okay. We had 5 nests, and 40% of those nests had blueberry pollen. Not a bad story, but not a strong return on our bees. In 2024, same story, bad return, but we put these in Oregon on a monoculture, and they did collect all blueberry pollen all the time. So another complex story. Here are the pictures I got of blueberry pollen. As you can see, it has like this, I think Kelsey calls it like a Mickey Mouse shaped pollen. I think it's quite a fun one. All right, so for this one, the main issue we're having with Osmia ribiflorus is dispersal. They seem to really like the blueberry pollen, really collect it, but they're not staying at the sites. And so what's the point if you're gonna lose all your bees in 1 year? So we're hoping that perhaps we can use Teresa's Genius lure next year, put that on the nests and see if we can get ribiflorus to stay. The lure hopefully should work for all sorts of Osmia species, so we're excited about the prospect of doing that next. Okay, raspberry. This is kind of a quick one. This is raspberry pollen, and that's alfalfa pollen, and that's sweet clover. I can't tell the difference. Sweet clover looks a little different from some angles. Some angles it looks very similar. All of my fields were surrounded by alfalfa, so I have a very hard time telling you for sure that any of the nests that I see have actual raspberry pollen. I want to be conservative and smart with my estimates here, so potentially I would like to use molecular methods for some of these samples, especially ones during peak bloom for the bees that I'm really thinking would be fans of this crop. So yeah, for now, no solid results because I do not feel confident IDing that pollen. Lastly, strawberry and Osmia lignaria. Strawberry is also kind of a not unique looking pollen, but it has this very interesting fingerprint-like texture on it. Unfortunately, I had a really hard time capturing this in photos, but you can trust me when I look at it with my eyes. It has this fingerprinty texture. It has the 3 apertures on the side. And this year, I found some really interesting things in a strawberry high tunnel in Utah, where we had 9 total nests, and 44% of those for Osmia lignaria collected strawberry pollen. This supports previous literature that study where they put mason bees out in the field, but it's really exciting to see them do it again and to see them doing it in a high tunnel. Okay, so key takeaways, pretty simple. Like I posited in the very beginning of this talk, I think we should use a diverse set of bees for a diverse set of crops. I think we can probably get better pollination if we're using bees that visit similar wild relatives in their ordinary lives. I think also using a diverse set of bees is more sustainable, less risk of all of your bees failing to disease or pesticide sprays, things like that, if you have all your eggs in different baskets. Also, osmia are not just orchard bees. We can use them outside of this traditional framework, and I think they can do quite well. And lastly, pollen ID is a really useful tool for bee management. It is not necessarily a useful tool for determining pollination networks out in a field, but I think it is really useful for management specifically. All you need is to find a used microscope and some dye, and you can do this yourself. And I think it is really useful and important because you need to know that the bee you're putting out there is actually pollinating what you want it to. It's really useless if you just see it nectaring and you think it's pollinating, but all it's doing is just stealing resources. So I think this is a very valuable tool. All right, that's my talk. If you have any questions, you can ask me now or email me. These are some funding sources I have to thank for my work. Yeah.